Okay, let's have a class and just an announcement. You guys gonna have a group for the presentation. Start from Thursday. I'm going to have a sheet for you to fill it. Okay, 2 to 3 of you. Not everybody. If you insist that you really want to do individuals, that's okay. It's not a problem. And we will start from April 11th. We'll have 3 classes for the presentation. Then follow the last 2 classes will be the lab section. That will be moved from the lab. We'll do the gram stem and we'll do the spread plating, pore plating, let you have some practices. Exam 2, the grades have been put onto the eCampus. Some of you did really good, some of you didn't do very well. You can redo after the final exam, during the final exam week, okay, or later. So let's continue to talk about this section. We will finish talk about the replication, transcription, and translation today, relatively brief. Because those 3 classes, if a genetic class or my major microbiology class, that took for 3 or 4 class sections. So we're not going to be doing that much. So we're going to talk about this DNA replication first. So we finished talking about the structure of the DNA, all those basic information, and we know the DNA replication is semi-conservative. And during the history, with the development of the genetics, all we said about chemistry back in 1960, there is 2 great scientists who did a very magical research to confirm the DNA replication is semi-conservative, which is Matt Mendelson and Star Research. Okay, Mendelson and Star Research. Those 2 are great scientists in Caltech University. When they do that, they are graduate students. So how they did it? I have a DNA. Okay, I'm gonna be labeled it. I don't have to do the original one. I don't have to label because majority of these are N14. And I use the DNA from the bacteria. So we're gonna see the bacteria will be growing there. I will let the media, which is showing you is N15. Then what's going to happen? The double bands will be open, and then you semi-conservative based on the gene complementary policy. You have this N15, and the original bonds will be N14. And we will say these is isotope, and you know that. Okay, what is an isotope? Molecular weight, slightly difference, chemical symbol is the same. But there's no radiation there. We don't have to do it. Okay, now what is going to be the next stage looks like? Next stage, we're going to have to find this guy. Is that right? We're going to separate them. So what happened? This one looks like that. This is N15. And this is also N15. And these original 2 will become N14 and N14. And on they do. What happened? At the end, you will see most of them, I will say more than 75%, is composed by N15, and less than 25%, even more, much less than that, will be N14. So what they did? They do a centrifuge, which is gradient centrifuge. And then you add a Sicilian chloride. So what they can find, the majority of these new bands, which is at the middle, at the bottom, is N15. And the top is most likely is M14. So this is from a chemistry standpoint, which is confirmed the semi-conservative policy or mechanism for the DNA replication. So we're going to talk about the DNA replication. Okay, what is that? For the DNA replication, you learn in other classes. Some of you do not have a microbiology, biology background, so we're going to explain to the way relatively brief. Since this is a microbiology class, so we're using bacteria as an example. This bacteria typically is E. coli. Okay, in your biology class, you are using fungi or using eukaryotes. That's a little bit different. So for bacteria, the replication happens is at replication fork. That's a circle places with the replication fork. It's a Y shape. And originally, you know, because it's double helice, so the two strand is compound very well. So I need something to start it. Okay, at the start, we have a DNA group, protein group. This encoding then A, then B, and then C, which is DNA C. Then A, this is, we call it the initiator, which means the places we started to have replication. Let's say get everything ready. Then B is a helix. So helicase. Why we need a helicase? Originally is double helix, and we have to break down. So we need an enzyme called helicase to break it down. And this helicase, we need a holder. So we need a DNA-C, which is the delta C. And the detail of that, you can see right here. Helicase, okay, delta B. Delta A is replication, sorry. See, you can delta A is replication, and delta B is helicase, and delta C is a holder, loader, okay? Which means a helper or helper. I'll just say assistant. Now, once these double bonds has been separated, because they are highly twisted, so it's very easy to come back. So we need something to hold on them, let them stay separate. Therefore, usually we have those protein is attached to these separated double bonds. This is called single strand protein. That's called SSP. We need those to keep them separate. Okay, let's say everything is ready. We got to start to do the replication. Okay, I have two bands, 3' to 5', and 5' to 3'. And I'm going to draw a little bit of sequencing. The 3' I always going to draw like this, OH, like A, P, T, P, C, P, G, P. Let's say A, P, T, P, C, P, and G, P. And remember, regarding the DNA, this is the DNA structure. This is the base. This is H, deoxy. And this is H, this is O, OH, that is 3', and 4'. Then you have CH2, and here will be a phosphate. Somehow like that. Okay, so we always write down here is from the 3' we have a hydroxy end, and the 5' we have phosphates. So we write it like that. Now, the top band, we call it a leading band. So why it is a leading band? Because we could do the replication automatically, or we say it's naturally, based on gene complementary policy. So what's going to be looks like? That will be T, there will be A, okay, there will be G, will be C, there will be T, will be A, this will be G, will be C. And obviously, you have so many of these free available gene base, ATGC, which is carrying a triple phosphate and ready to come in. And these two phosphates usually will be hydrolyzed. Now, and always remember, even this 3' to 5' is a leading band, they still need something to initiate. We call it primase. This is usually a very small RNA sequence to do it. Okay? RNA sequence to do it. So let's say we do it. Now, DNA polymerization. Since we already call it a polymerization, obviously we need an enzyme. This enzyme called DNA polymerase. Now, what is the DNA polymerase? This is the one to do the polymerization. We have DNA polymerase 1, polymerase 2, polymerase 3, 4, and 5. We basically, here we are talking about DNA polymerase 3. They do in the polymerization. Now, who is the person who find it? This is Arthur Cronenberg. Arthur Cronenberg in 1960s found it and got the Nobel Prize in 1970. But there is a joke about it. It's not really a joke. It's ironic stuff. What happened is the Arthur Cronenberg find the DNA polymerase is actually not a major polymerase. It is actually a small sequencing of the RNA. But it still contributes, the credits given to him. He's find the polymerase. Okay, so they do the polymerization. Now remember, the polymerization is always 5' to 3'. Why? There are two reasons for that. You go here, okay? Then you go here, you connect it. Another one, let's say T, H, 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 this is O, this is CH. Okay, that's 3'. You can go here, this is 5' to 3'. Because we always have this OH. Can we do the other one? No, it's not working. Why? And you learn in your other classes, you look at the structure, you know, because 3' to 5', no available free hydroxy bonds. So you cannot do on the other one. Okay, this generates a problem. We said the DNA polymerization is always 5' to 3'. Then how come the other we do? How are we going to do it then? Okay, we have always do like this. We're going to have to do like this, like this, like this. No, it doesn't work. So, how we have to do it? We need to generate those small segments. Okay, those very small segments. Those small segments has to be leaded with an RNA primer, and those small segments called Okazaki segment. This Okazaki segment is coming from a Japanese scientist. So we have to have those small, very simple Okazaki segments to have a small piece of the DNA. Then we have to cut these RNA primer. These are RNA primer. We have to cut it. How are we going to cut it? We will need to using DNA polymerase 1. Okay, so we have to cut them. We cut them, then we left on some of the space there. Okay, we left lots of space. What we have to do? We have to seal it. This process we call it a ligation. And luckily we had an enzyme called the ligase. Then we can connect it. Okay? That's what we have, 5' to 3'. Now, the way we have to do is 5' to 3', 5' to 3', which is opposite. Okay, go the other way. So that's something we talked about. But at the beginning, when the DNA created, a new double bond is created, there is some problem we have to solve. The first problem, topological issue. Because with a new synthesized DNA, it is most of the time is positive supercoiled. Positive supercoiled is not easy to separate and not easy to be functional in the next step. So there's a higher tension. So we need to release this tension, become negative supercoiled. So what we need some other enzyme. This enzyme called topoisomerase. There is a couple of topoisomerase. There's isomerase 1, isomerase 2, 3, 4. Topoisomerase, specifically in E. coli, we call it DNA gyrase. So that's the first issue, topological issue. Okay, topological issue, sorry. Second issue, fidelity issue. Fidelity issue, which means how accuracy it is. When you have a new bonds generate, how accuracy it is compared to the old mother bands. There is also some question about it. So naturally, if we do not do anything, the new bond, there is an arrow, could be as high, 10 to the minus 3. That's very high, and we don't like it. So we have to do an initial proofreading, rely on DNA polymerase 3 to do the proofreading. Using proofreading, the arrows could be reduced to 10 to the minus 5, 10 to the power of minus 5, 10 to the power of minus 6. But is that going to be good enough? Sounds like it's a very low chance of the errors, but it's still pretty high. So we need to further to do it. There is a methylation methods, which means if I have ATGCG, I have TA, GC, let's say AC. What I'm going to do it, I'm going to add a methyl group right here. Then this will be cut down and come back, add a new corrected G there. So we can do a methylene group. And this will be reduced arrow to 10 to the 7th to 10 to the 8, minus 8 even to 10 to the minus 9. This should be fulfill the requirements of the accuracy. Now, more or less, we have some mutations will happen. Okay, then the system have to do more jobs. If really something life-threatening, then the system will have SOS recovery system that's engaged DNA polymerase 4 and 5. Okay? So, this is pretty simple, pretty straightforward. We talk about the DNA replication. And we have a slides for that, you can look at that. And we also have a slide which is talk about the function of those enzymes. And you don't really have to remember them because we do the open book. But I do notice that some of you even do open book didn't do very well, so you have to be really pre-read some of the material. Make sure you at least read through it before you go for the exam. Okay? Otherwise, you're going to spend tons of time to find the answer. So this is the first part we talk about, is DNA replication. Now remember, we were talking about the principles here is called the central dogma. That is DNA replication. We finished. The next step is RNA transcription. DNA transcription, sorry. And the finally is translation. So we want to cover these two later, okay? So let's talk about transcription. When we talk about the transcription, we need to mention about something. You had RNA gonna jump out, and this is DNA. So we want to talk about what's the difference between them. What's the difference between RNA and the DNA? The biochemistry class could be long and long, listed lots of difference, but here we'll be briefly mention several important things could be the characteristics of the difference. RNA could be right here. So what are the difference? Same thing, you have a 5 sugar carbon. This is the base. Okay? And this is the phosphates. I'll just draw very simple. Same thing here. 5 carbon sugar, a base, and the phosphates right there. What's the difference? The first difference. This happens right here. In the 2 prime, I don't have O. That's why the DNA full name is 2 prime deoxynucleic acids. And at RNA, I have an O there. So, this is ribose. For the RNA, DNA is deoxyribose. That's the first difference. Okay, the second difference, we talk about the base. For the DNA, it is adenine, thymine, cytosine, and guanine, ATCG. For RNA, ACG stay the same. What's the difference? Is uracil replaced thymine. So what's the difference between that? This is, we talk about a couple of times. We talk about this a couple of times. We have this guy right here. So we have a CH. Is that correct? We just draw it. That's a thymine. What's a uracil? exactly the same most of the time. You can just draw it. Some people say in the microbiology, the biochemistry class, say the drawing these things are difficult. It's painful, but actually it's not, because the majority of these are the same. Okay, so we'll have this guy here, this guy here. Now what's the difference? If you learn it in your chemistry class, if you don't learn it, so it's okay. 1, 2, 1, 2, 3, 4, 5, 6. Carbon 1, 2, 3, 4, 5, 6. This is difference. What we call it. So uracil at carbon number 6, instead of you have a methyl group, you have a proton, you have a hydrogen. And we say the hydrogen is at an unimportant place. Now because of that, which makes U relatively unstable. So we can say RNA relatively unstable and DNA relatively stable. Now last thing, everybody knows about it. RNA is most of the time single strand and DNA is double strand. Now another thing is that for the DNA replication, there is an arrow fixing system. We talk about that just. For RNA transcription, there is nothing to fix it. Which means if you have a mistake happened in the replication, all the way will happen from to the translation. This will cause molecular disease like that. Okay, so this is something we want to talk about before we do. Now, what is transcription? Transcription, this happens on the DNA double bonds. And I have 3' to 5', and also 5' to 3'. Transcription does not mean you just find any places you start to do the transcription. Now obviously the transcription is also from 5' to 3', there's no question about it. They need something to assist it. What are they? Number 1, same as other biochemistry reaction, the transcription need an enzyme. Because it's still a polymerization process, so transcription, instead of we have DNA polymerase, we have RNA polymerase. And all RNA polymerase is very interesting. This is a hollow enzyme. So what they have, the hollow enzyme has alpha, alpha prime, beta, and omega. This section we call it a core enzyme. And then they need a cofactor. The cofactor is sigma factor. And then most of the time, we call it sigma 70. Why is sigma 70? Because it's 70,000 delta. The molecular weight is very huge. And that sigma 70 is responsible for most of the bacteria normal life. So we need RNA polymerase. But the RNA polymerase is not just to find something to start to do. They need to recognize a very special sequence. The special sequence has been a lot of the research to do. A very common one has been reported. Let's say we start the transcription, we write them plus 1. Okay, on the right side, we call it a downstream. And the left side, we call it upstream. Around the 10 BP base, there is a very conservative sequence called T-A-T-A-A-T, complement with A-T-A-T-T-A. This is called a minus 10. We call it pyramidal box. Okay, then we have some space between that. It's 16 to 18 BP base pair. Then around here is minus 35. We have another sequence. Is TTGACA. I need to make sure what I'm writing is correct. Because I was teaching TTGACA. I was teaching for chemistry. Lots of chemistry. I was writing on the whole blackboard. So, okay. Corresponding to AACTGT. This whole area we call promoter, and the RNA polymerase have to recognize it. Okay, the RNA polymerase recognizing it, they started to do the polymerization. So for RNA transcription, the key point is not how they do, it is really at the beginning. And I guarantee you this slide is important, you can mark it. You will use in the exam. Because this is well tells you the promoter, the region is minus 10 and minus 35. Now, how they do the polymerization, this is what you should know. Okay, I have a double bonds right here. 3' to 5'. 5' to 3'. ATGC, ATGC, just easy. Okay, what is the bands of the second one? TACG, TACG, you know about that. Okay, what's going to be the sequence of the mRNA? 5' to 3'. Don't forget about it. What's going to be it? Everything is exactly the same to the top bands. The only difference is U replace T. So GCAUGC. Look at here, that's the only difference. Therefore, the top bands, we call it coding band, and the bottom one, we call it a template band. Now, how do we know the transcription is going to start? There is a certain mechanism. You see those hairspin area with rich U. That means you have to stop it because it's unstable. Remember we talked about this. Uracil is unstable. And sometimes you see the sigma factor, or we call it a rho factor, then you stop it. And there's some other mechanism. Okay, we just be relatively brief around here. So you can see the slides right here. That's rich U area, hairspin, then caused by, that's a replication what happened. Or that's transcription what happened. Okay, the next one. What we are trying to do is translation. Okay. This brings you something relatively new. So we're going to talk about this translation. So take out this transcription. So what going to be the translation looks like? We took this out. This is a very interesting terminology. Okay, why we call it is transcription? Why we call it is translation? That's a question I asked at the beginning of the semester, even the beginning of the section, if you remember. What means translation? What means transcription? What's the difference? It's kind of very interesting terminology. Translation and transcription. This is letter. Sorry. So this is letter, DNA sequence. Is that right? We have ATGC. I'm going to DNA transcription. Will become RNA, mRNA. Let's say it's still letter AUCG. But how about translation? It becomes 3 letter, but they don't become a protein. Well, of course, they have amino acid polypeptides, which is a modification become a protein. So what's the difference? This is letter to letter. This is letter to protein. That's called a translation. That's the difference. Transcription only letter to letter. Transcription, translation, letter to protein. That's what the major difference between that. Okay, it's like an English transfer to Spanish or transfer to Chinese, which I speak before I came to the United States. So this is a picture which is showing you how we're going to do it. Now, before we do this, we need to know. During the transcription, we rely on RNA is mRNA. We also call it a messenger RNA. Because they carry on the 3 codon. We'll get to that very soon. Since we talk about the translation, there's somebody have to do the transfer. So we're going to have this guy comes out, which is transfer RNA. Now, what is transfer RNA? It's a very special structure. When you first see it, it's kind of very interesting. Look at this, what it looks like. Kind of very funny. We call it a clove leaf shape. Clove leaf shape. Leaves shape. There are different arms. What are these here? They're always a tricky question for the biology. When you go biology 101, what's tRNA at the bottom carry on? If this is the mRNA, you have AUG. What is the tRNA? That's UAC. So that's an anti-codon. That's very interesting. It's not codon, anti-codon. It's called codon, so anti-codon arm. What is this one here? That's called a variable arm. What's right here? This is called the T theta C arm. And they have a PY, PX. So how do you know? If I give you a sequencing, it is tRNA. You just run a whole genome sequencing. If you see there is a very special gene base, like a PYPX, in addition to AUGC, then you know it is tRNA. That's another tricky question in the exam. How about here? This is DR. So, first of all, you need to know what the tRNA looks like. And that's a picture we should show you. It's very, very interesting, to be honest. Now, we want to talk about this. This is very interesting. And we talk about amino acids, then going to polypeptides, then going to protein. There's lots of things we can talk. Okay, that's a couple of classes for biochemistry. Now, amino acids, we need the letters. How we do the letters? 3 boxes. We name them box number 1, box number 2, and box number 3. Okay, I have a very simple math question for you. Everybody pretty much know. How many gene base could express amino acids? A, U, C, G. So theoretically, how many different codon could be generated? How many possible in box number 1? 4. How many possibility in box number 2? That could be repeated. 4. How many in the last one? 4. So how many possible? 64. 4 multiplied by 4 multiplied by 4. But how many codon we have? 61. Now where are those 3? We have a stop codon. Is that right? We have a stop codon for that. So what are they? UGA, UGG, and UAG, if I remember, the stop codon. Yeah, UGG, UGA, and UAG. That's a stop codon. Now, let's all have another question. We talk about, during the replication, they more or less have some mistake on happening. I always say this is like do an exam paper. You barely do 100%. You'll be very happy when you do 90%. Same thing for bacteria. So let's say there's a mistake happen. Replication, transcription, or translation. They're going to cause problems sometimes. But every bacteria has to survive, same as a human being. What they do? I have box number 1, box number 2, and box number 3. The biology is very smart, all those stuff, especially bacteria. They need to be a very strict for the box number 1. You have to make sure it's correct. The box number 2, you also need to make sure it's correct. However, box number 3, I gave you flexible. That called Warble impacts. Or Warble impacts. Therefore, I had 61 codon. How come they could be express 20 amino acids? There is some redundancy happens. There is some regeneracy happens because of the Warble impacts. Okay? So if you look at here, you look at this one here, it's very interesting. You could see phenylalanine, for example. UUC is phenylalanine. Serine, UCU, UCC, UCA, UCG is phenylalanine. Leucine, CUU, CUC, CUA, CUG. Look at the last codon. The box number 3 is gave you flexible. The box number 1, number 2 is always stay the same. So, if you have some mutation happened at the box number 3, it may not affect you at all. And this is called silent mutation. Because it's very quiet, it's not going to affect you. Okay, so this is something we want to talk about before we go further. Now, we're going to be how they do it. Okay, you need to know the tRNA structure. You need to know how they connect. This some of the picture looks like a little bit complicated, but it's always simple. We draw a brief picture. 5 carbon ribose. This is the gene base. Okay, always remember this is the OH. Okay, this is the H. Oh, we needed it. Don't worry about that too much. CH. CH. CH2 goes here. Phosphates. Where are they gonna go? This is gonna go here. Prime and 3 prime. They connect. And this is tRNA. Now where is Kia gonna go? How are they gonna connect it? Amino acid group over VSL. NH2, long and long. So, what is the basic structure of amino acids? Center, chiral C. Proton always have. Amino group, carboxy group, or side chain. Is that right? Right here they connect. This is a tRNA. And always remember, the first amino acid, it is always MET. Methionine. That is, what is methionine? CH3, CH2, CH2, then go middle C, then go H, then go NH2, and COOH. That's methionine. Now, specifically for bacteria, it is a little bit different. Bacteria, the first one is always very special. I'm just to show you. That's called formylmethionine, a little bit different. Okay, you can use its methionine. It's a little bit special. Okay, so this is something we want to talk about that. Now, so we're going to go to the process. How are we going to do the replication? Now the question will be back to you. Where going to happen the replication? Where will happen replication? Replication have a very special location where it is. It's a ribose. And you always remember, it's very magic. Ribose is a very few cell machinery both prokaryotes and eukaryotes have. Because it's important for replication. For transcription. For translation. How we do it? There's the two subunits, okay, originally. For bacteria, 70S. Top, big one 50S, small one 30S. Okay, they never add up together. Why? Because the S is Vandenberg units, which is testing the speed of precipitation. It's not your molecular weight. Okay? So always remember, not the simple mass. It's the Vandenberg units. The Vandenberg unit for precipitation. So at the beginning, the 30S and the 50S, they are actually separated. Now when they separated, of course they need something to make sure they're separated. So there is our initial factor 3 goes there. Okay, now what happened is the first step. We talk about the tRNA right here. I'm going to carry on this guy coming. So the tRNA is going to come in. Okay, they're going to carry on. AAA. They go in. How they come in? They not come by themselves. They need a boat. Or they need an Uber driver to take it there. What is that? Elongation factor 2. And once this happens, this guy gonna go. We need the elongation factor. We need initial factor 1. And this guy gonna come down, so it becomes like this. Now obviously, you come here. You go any places, you need to pay the tickets. Unless it's National Park, West Virginia State Park is free. Very few places. But if you want to watch a football, you go studio, you have to pay. Okay, this is need energy GTP. Hydrolyzed become GDP. So that's the initiation. Now we're going to start to do the biosynthesis of those three codons. It's not like you just go there and you start it. Everything is designed very smart. So I go here. Okay. I have this comes out. Then you have to recognize a very special sequence. And that very special sequence is easy, called AGGAGG. What's the name for that? SD sequence. QUENC. That's called the Shai-Dagano sequence. Dagano sequence. And that Shai-Dagano sequence has to be recognized a very special material at the 30th subunits, which is 16S rRNA. Therefore, you need to know the RNA. There are three different types of RNA. mRNA, message RNA, tRNA, transfer RNA, and rRNA, ribosome RNA. And the ribosome RNA is actually a subunit of a ribosome. Okay, once this guy happens, they will start to do the synthesis. This comes out with three major boxes. EPA. This is not Environmental Protection Agency. Okay, there are three locations. What is E? Exit site. What is P? Peptidal site. What is A? Amino acyl binding site. So let's assume there is a tRNA comes here. This is AA. Okay, MET always at the top. But the first one. Okay, I have a second one coming. How they do? This guy always goes here. So the next step will becomes, let's say EPA. What will happen? AA1, AAMET. And they go this way, and this is the free one flee away. The tRNA go to carry another amino acids, then come in. So this come in here, become a new one, AA2. So it goes here. And this is the molecular, is always go here. They come long and long, long and long. And here, then CR2H NH2. Go this way. And also 5' to 3'. Now people never understand why this happens. When you read the textbook, flip it over. Now, our slide's actually pretty good. This is upside down. Can you see it? Lots of the textbooks, they draw like this. You're never going to understand it. When you understand it, you have to flip it over. Because that's 5 primes, that's 3 prime. That's 5 prime, so go this way. When you flip it over, you understand it. Okay? So that's what the synthesis is. What's the synthesis for? It's kind of the very interesting material for that. So let's go for see something new. Now, there's an elongation factor. Once this happens, there's an elongation factor. And finally, the chain will become long and long, and they will be released. When they release, they also need something which is called release factors. Now, the whole time, they need the GTP, the hydrolyze, to do it. Okay? So that's the whole process. If you do not understand it very well, you can look at this picture. This is like a cartoon movie picture, which is explained very well how the translation happens. So I draw here, which is briefly introduced to you, But I would love to go over these slides to you a little bit. What are they talk about? Okay. This is a very nice picture, which tells you the translation, the replication, transcription, and translation. Be careful. The exam question is which ends. Because one is rounding 5' to 3', another one is 3' to 5'. The opposite direction. And the AEM is also 5' to 3'. Don't forget about that one. And you always remember that TRNA is carried on the anti-codon, it's not the codon. The codon is mRNA. This is the one I always talk to my major students. You don't have to remember this. If they need it, I'll give you a sheet. But always remember the first one is methionine AUG. And the stop codon is UAA, UGA, and UAG. What this tells you, in other words, which means if replication has one misrepair happen, they're going to follow into transcription because no fixing, and then the translation. If the mistake suddenly happen, become a stop codon, then the translation will be stopped. That will be dramatically change the bacterial function. Which means originally I'm trying to have 100 amino acid sequencing. All of a sudden it stopped. It become only 20 amino acids. Of course it's going to bother the biological function. Okay, so be careful about that stop codon. Sometimes it gives you big trouble. This is the one which is gave you a very good example. Explain what the wobble force. They give flexible of a third place. This is I already talked about the joint structure of the tRNA. Like I said, there is some of the unknown, very weird gene bases. A couple of arms. And this is the structure, which is like here. But I don't have my hands to be that high. Textbook is upside down. Okay? A little bit different. This is, the transfer RNA. Always remember the ribose is a place, had a translation, and they never add up together because of Vandenberg units. 16 sRNA binding sites, Shide-Agano sequence is AGG. That is usually the 30th subunits. This is where it comes when you go WVU Health Science Center. You do research with some faculty there. They always say 16th RNA biotechnology. That's where that comes from. Okay? Now, obviously, the 50th subunits, we have 23 RNA. This is always very special for a bacteria. Bacteria start from formyl methionine. Fungi, eukaryotes start from methionine. And this is the structure of the formal methionine. This is the one from another standard point talk about the initiation of the translation. And they need an initial factor. The Shide-Agano sequence recognizes the 16S RNA. And this is the procedure how they do it. I briefly draw on here. In my major class, I draw another 30 minutes to talk about the detail. And elongation, and the needed elongation factor. There is a 3 location EPA, exit amino acid and the peptidal site. And when they stop, they need a stop codon and they need a release factor 1, 2, 3. Now, always remember, the whole process needed the energy currency is not ATP, it's GTP. So that's why what I'm saying, it is important to know ATGCU, they are all could be an energy currency. It just depends when they use, but ATP is most widely used. That's picked by randomly by the bacteria. And this last one, if you like it, or you never know, it's like a cartoon movie about it. Now, most of the time, if you really never taking any biology class, you still don't understand, go to the YouTube channel touch DNA replication, transcription, translation. There are lots of the cartoon videos there. I know lots of the people, they broadcasting like 20 minutes. I don't do that. You can watch at home. It's a lot of there. Okay? So this is what we have is for this section. Okay, so very briefly, we talk about the DNA replication, transcription, translation, which is the central dogma. On Thursday, we will go another section, we'll go over the biochemistry, which is talk about aerobic respiration, the calculation of the ATPs. I will skip the structure of the molecules, but the name we will mention. In my major class, we talk about all the structures. It's a tedious blackboard from the top to the end. Okay, but here we're going to be only mentioning about the chemical names, and we will skip about that. Okay, now last thing don't forget is your presentation. You need to start to gradually prepare it. And I want you to do something which is fun or creative instead of just use a just present the paper. I mean, if you have something fun, that will be a plus. Okay, so that's all I have.